بسم الله الرحمن الرحيم. أعزائي الطلبة، نرحب بكم. محاضرتنا اليوم بالفيروسات الطبية. أبنائي الطلبة، هرجع من الأوراق اللي دزيت لكم إياها، فأرجع بريف ومختصر حتى تصير عندكم الصورة الواضحة عن المحاضرتين الأخيرة. أول شيء introduction. الفيروس, what is meaning by virus? Virus is a smallest unit for infectious disease. Usually viruses present in nature. Virus differed from bacteria from the following thing. First one, virus usually may be DNA or RNA. Second, virus not have cell wall. Third, virus can not growing in a synthetic media. Fourth, virus resist antibiotic during the treatment. From this, we can differentiate it between virus and bacteria. Virus usually contained of the following component. First one is genome. Genome, this meaning maybe RNA or DNA. Virus usually may be RNA or DNA, but not both. During the classification of virus, usually depending of the followings. Later on, discussing. Now we start the genome, the active form of nucleotide, nucleic acid, maybe ribonucleic acid, or maybe deoxyribonucleic acid. Usually present, for example, these as a virus. This is a genome, RNA or DNA. These genomes surrounded by some membrane this membrane called capsid. Capsid is a protein substances surrounding the active form, which is called DNA or RNA. The DNA or RNA with a protein called nucleocapsid. Nucleocapsid. This nucleocapsid is active form, which is called a viron. إذن الفيرون هو active form of a virus containing RNA and protein layers. Some virus, usually animal types of virus, animal virus, addition to the viron neurocapsid, we have another layers, which is called envelope. The envelope consisting usually by layer of lipid. Then this bilayer of lipid covering the viron. The viron usually is nucleic acid, which is called a genome, plus covered with capsid protein substances. In addition of that, sometimes we show it an envelope. Now we move for classification. Classification of the virus usually depend on the following. The first one, morphology. What is meaning by the morphology? Size, shape, volume. Size, shape usually is very important in the morphology. Second one is biochemical activities. This meaning thermo action that the virus when it has stability against temperature. Third, type of genome. Type of genome meaning either be RNA or maybe DNA. Others, proteins. These four proteins meaning a capsid, additional layer, usually other additional layers present usually with the envelope here. With envelope inside of envelope layers, we have some protein layers called matrix. Matrix, other protein layer usually present inside the envelopes. Here we can find this matrix. These layers not in all virus present, even though in the envelope, not all envelope containing these materials. Others, lipid. Lipid usually present in a bilayers in envelope. Carbohydrate, 6 carbohydrate. Carbohydrate present the main constitution in the outside of virus, usually is glycopeptide. This glycopeptide covers the outside of virus body. And later on we discuss this function of these layers. Finally, antigenic properties. Antigenic properties is very important to all virus. When virus infected animals produce antibody, these antigenic properties meaning ability to produce antibody. From all these, according to classification, usually virus may be DNA and RNA. In RNA usually is single strand RNA. In DNA, double strand DNA, or sometimes single strand DNA genome. The other properties of the virus is a sense. In general, virus divided into sense types. Sense positive, sense negative. In the sense positive, meaning have usually a messenger RNA. In a sense negative, need complementary RNA dependent DNA. These the main differentiating between sense positive rather than of sense negative. Now we move for the shape of the virus. Shape of the virus usually present in three forms. A, helix, B, icosahedral, C, prolate, and D, complex. The form of helicals, from the meaning of helix, usually present this form, meaning a cylinder Other types is icosahedral, meaning مجسم معناه present usually like here of a virus. This is head present in icosahedral, containing many thousands of units. These units names as capsomer or sometimes protometer. This is sometimes virus as a head of virus, sometimes or species rather than head and tails. Virus usually infected both plants and animals. A plant, how can affected by a virus? As we know, a plant have a rigid and thick cell wall. Is difficult to penetrate by a virus. Usually, virus choosing a pores between the cell wall of a plant. By these pores can introduce to the vegetative cells of a plant and causes a disease. In the animal, there is many routes for transmission of a disease of the virus. Sometimes by inhalation, a droplet of sneezing, cough like flu, corona, or by blood like the hepatitis C, or by sexual activities like AIDS, HIV, or by fecal content when contaminated a food or water and take it internally. By this route, different route can cause a disease of the virus. When the virus introduce the mechanism of the virus, how to introduce the cells. First one is attachment. Virus attached the cells, as we mentioned in previous time, that the outside of a virus contains a glycopeptide. These glycopeptide have specific receptors in the cells. For example, the AIDS disease, the virus, when infected the white BC cells, which is H cells, helper, have some receptor site outside of the white BC, which is called CD4. C meaning cluster, deaminase. These CD4 are present here in the lymphocyte. This is usually as a receptor for specific virus, and for example is HIV, human immunodeficiency virus. When attached the virus here or here, these called the attachment. Virus active form usually is viron. Viron is the active form of a virus, usually is RNA or DNA and this capsid. Second is encoding. When the virus attaches a specific receptor site in the human cells, have ability to introduce inside. How? By fusing together with the cytoplasmic membrane here, causes a pores. During the spores, start primarily by uncoated, meaning the capsid is disintegrated. Capsid is removed. Only genome introduced inside the cells. These now present in cytoplasm. In cytoplasm, usually, if is DNA virus, genome went away toward nucleus. If is RNA virus, genome went away toward cytoplasm. Attachment, uncoated, and third, transcript. During transcription, start multiply the virus by join the RNA with the cytoplasm and getting by RNA polymerase enzyme and start to produce messenger RNA and later on protein. During the formation of a protein can be specification of protein to two forms, maybe RNA or DNA or origin protein. From these start again RNA or DNA with the capsid protein, a new virion produced. And infected another cells. In bacteria, virus infected produce two forms, lysogenic lytic and lysogenic form. For example, here a bacteria, virus attached, then introduced and start to multiplication, and later on ruptures of bacteria and getting bacteria outside. And lysogenic, here bacteria, and this is plasmid or ribosome, integrate RNA or DNA with integrated with plasmid or ribosome. During the multiplication here, forming a new meaning produce a new and be silent division, meaning not causes damage like alighting the bacteria. From all this lecture, I mentioned and explanation and briefly the virus. I hope to see you in next lecture. Goodbye.